Welcome everyone to Immuno-model first webinar, Emerging Innovations for Immuno-models. And we are excited to host representatives of two companies that will present their systems to model cellular and tumor microenvironments in vitro. So we will start with both talks and followed then by a question and answer session. Please write your comments and questions to Q&A session, which you can find below at this Zoom platform. So the first speaker is Professor Krzysztof Wrzesinski, which is a co-founder. He's a co-founder of CelVivo and professor at Faculty of Health, the Northwest University, South Africa. Krzysztof Wrzesinski obtained his PhD from University of Southern Denmark and has a strong background in biomedical science, having worked with research for the past 25 years. His work focuses on advanced cell culture technologies and development of drug testing pipelines based on in vitro models. Today, we will listen about ClinoStar, a purpose-built system for culturing cells as in vivo mimetic 3D structures. So, Professor Wrzesinski, the floor is yours.
Thank you very much. I hope you can hear me well. And thank you for a very nice introduction. And I will now try to see if I can share my screen. So I hope now you can see my presentation. Sorry for that delay. It's always like we did the try and it was working, and now of course it kind of gets a bit delayed. So once again, thank you very much for inviting me for this talk. I hope I can kind of bring a small inspiration to the audience. And I would say yes. I am now a chief scientific officer of a company which I actually became I am a co-founder of it, and the company is basically a spin-out of the university, where we, as two university professors who were actually trying to do our experiments, and we have figured out that we need a piece of equipment which we cannot buy. So we say, all right, let's do it ourselves. So that's how it started in 2014. Actually, we have created a company with a capital of 1 Danish krone, so it's 0.7 euro, let's say like this. But what we did, we actually were able to build in our garage basically a machine which was fulfilling our requirements. So we need a machine which will be able to keep cell culture alive for a long period of time, and then the 3D come on top of it. To come to the long story short, I would say that the company was slowly progressing until 2018, where it became obvious that we have to decide: are we pursuing our commercial careers, or are we staying at university? We decided to go with the commercials, and then in 2021 we actually launched our first very commercial machine. So the difference between the machines you can see right now and the ones which are the historical ones was that the machines right now are fully autonomic and contain everything what you need, where the previous ones were kind of insert to the incubator. I just want to mention that in actually 2023, means a week ago, we actually have launched a version 2 of our unit, which has some improved functionalities, and I will maybe talk a bit later about this. So why we did this? Because this was a very simple, and I think everybody is standing behind such a principles that when we build models, we would like to have them representative of physiological environment. We would like our models to be reproducible, and of course, because it's a model for our tests, we would like to have it abundant and accessible. So Another a general statement would be that from when you come from a monolayer culture to a three-dimensional culture, or from cell culture to a tissue culture, rather, we should maybe a bit change our thinking from the standard how fast I can have my cells ready for an assay, to how can I always have a reliable and relevant assay ready. That's more like a thinking where you work with the experimental animals. You have to prepare your animals for the experiment, but once you actually have your preparation pipeline ready, it does not prevent you from having ready animals every day. So that's what we have to think about our tissue culture. Of course, we would like to have a higher correlation with an in vivo situation, and one which is very important as well, that the system should not be overcomplicated, that the system should allow you to concentrate on the biological questions, and it should rather be a help and not a thing you have to master by itself. So we developed a system based on the ClinoStar principle, and the system contains two parts. One is the drive unit. It's a ClinoStar. So it's basically an incubator with all what you need for creating a creation right of an environment. And then you have also an disposable clinoreactor. So this is basically a cell culture vessel where you grow cells in. I would say if you will need more details about our what you exactly have and what type of beads and what type of materials, we have this on our webpage, and I would also be very happy to answer those questions. But I would think that it's much more important to show you what you can do with the system. So the system is actually, we say it's easy to use, but it's enable a long-term culture. It's actually scalable, so it's actually based on the cluster idea, that you can actually add more of the units to it. It gives you an uniformity of your model. It has a lot of features which allows to a remote control of your cultures. And it's also a closed system, or semi-closed or cartridge-based system, so it allows you to minimize the risk on infections. And one very important fact, which I will discuss a bit more later on, is the system is not producing an increased shear force in order to supplement your construct with the nutrients. So here you will see a small glimpse of our own lab, where we actually have a cluster of our ClinoStars, where we do our own experiments. So we use the clinostatic effect, so basically rotation, which allows us to balance a gravity force. And we can actually work with 6 different axles in each cube, in each module. And the module is also capable to control an environment parameters such as CO2 temperature and oxygen level. Here you can see basically how to work with the disposable vessel, which is prepared for both working as a manual operation, but it's the whole unit is also prepared for the robotized handling and media change. It's also very nice because you actually can use it for the imaging directly from the vessels without need of opening, so without actually increasing risk of the infections. And it's also a portal based, so basically anywhere in the world you can access your machine and see what's happening and actually adjust the parameters as you want. What we do with our system. So I would say first maybe we should go a bit to a basic of the 3D cell culture, and I know that there are many different divisions, and it's a lot of debate on how we should call the construct we are growing. So I just put here a simple slide, like we say, okay, we grow the organoids, we will be the primary or stem cells derived, we will do the spheroids, which will be the immortalized cell lines, and we also have a separate niche for the patent-derived organoids, so directly from the patient's samples. I know that some people say, okay, this is like a primary, but sometimes you can buy the primary cells from the supplier, and here we just want to mark that you can also basically take a donor cells directly from the patients and grow and work with them. And then we also have this tumoroid, so basically a patient-derived samples of a particular cancer. So if we just keep this simple division, I would say all of them are all of them grow in our environment, and all of them will basically be supplemented with a sufficient nutrient and gas exchange. If you think about a technology, so again, a lot of different technology is used for 3D cell culture. And what I could say, we can just divide them into two simple groups, which is in static systems and dynamic systems. Static systems give you a very stable environment, but it creates a depletion zone. So basically, you have our constructs don't have an active vascularization, so they don't have a pumping system, so they rely on diffusion. And in the static system, we have so-called passive diffusion, which basically limits the way how we can use it. It's a factor which we would like to overcome, and usually we overcome this diffusion depletion zone by basically applying a flow of media, so you can remove this zone. But when we apply the flow, we also apply a shear force, and excessive shear force is not something which we would like to have in our culture. So our approach is actually to create a flow, but actually create it with a so-called clinostatic effect or omnidirectional gravity stimulation. So where we use gravity to keep cell construct falling, and we use a rotation and the viscosity of the liquid to kind of move them up. So illustrated better, if you will look on this movie, you can see how the culture look like in the real life. Here you can say, okay, this is look like, and all things are very much spinning and rotating. But if we actually think about this and take this rotation out, if I will try to show it here, you can actually see this picture with the black frame. It's actually also rotating a clinoreactor, but we have electronically removed the rotation of the clinoreactor. So if you can imagine that this is something as you would now see as you are in a train, instead of as you would be on the station in the previous images. In the previous images, you just look from the outside to our clinoreactors, and it look like a lot of movements happen inside. But when you actually remove the rotation of the vessel by itself, you can see that the movement inside are very gentle, and we do not have much pressure difference, so we do not increase the shear force in our system. However, we have a very nice and uniform media flow, so we do not have the depletion zone. So this system is able to create a uniform low shear force environment. It's good for the long-term culture, and we basically try to mimic an in vivo functionality. Yes, how we do this? I would say we also do it with an ability now to control the environment with an oxygen level. You know that the different tissues in the human body have a different access to oxygen or different level of oxygen. So actually our system right now is also able to regulate that. So you are able to use a clinoreactor to create a different oxygen levels in your environment. And just coming a bit back to a standard CO2 environment, our system is extremely fast with recovery, and is also very stable. As you can see on this graph, it's extremely stable system with an CO2 and a temperature. Yeah, if you are interested more about the physics, I would say I'm happy to answer afterwards the questions, but also you can see our publications where we publish the physical abilities of the system. But what is interesting is also that we are very quick with a recovery when we do the opening and when you do the maintenance of your cells. But also when you think about oxygen sensors, and here is actually a picture from our lab when we were testing our functional models. But what is important is that the system is able to actually regulate the oxygen level from 2% to ambient regularly. We can also go to a 0.1% of oxygen, but I would say that will require a lot of gas pumping. So if the customer would like to have it, yes, we can do this. Our sensors are rated for that, but a standard level is around 2%. And what is important is that the speed of the recovery to the environment. So if you will see here, it is if we have a setup for 2% of oxygen, we need less than 45 seconds to actually achieve the set point from the time you had an atmospheric level of the oxygen. And what is also interesting that we use a relatively small amount of gases. Here I just give you an example that is around 800 liters of nitrogen per 24 hours when we are doing a standard operation with 2% of oxygen setup and 40 openings of the incubator. So just to say that we are also able to very quickly normalize the environment inside the liquid, inside our Kleino reactors, and we actually can very nicely keep it in the constant level. And opening of the incubator basically do not have an effect on the environment inside the Kleino reactor. So how you can work with the system? So first thing, you can basically do a single cell suspension. So if you would like to have a construct made out of your cells of interest, you basically take a single cell suspension and you place it into the incubator, and then the cells will do the self-assembly. This method is very nice, simple to use, but you will get a construct which might have not identical sizes. So you will have to create and procedures where you will actually pick the construct of the same size. If you would like to have a size, more control on the size or more control of the components of your construct, you can use this force aggregation method, where you basically use micro pattern plates, such like Agrewell or Kugelmeier or Euplasia from Corning, and you create so-called spheroid seeds, and then you grow those further in our system. You can also do the design spheroids, so a construct made with use of hydrogels. Here we have an example where we use encapsulation method with a sodium alginate on the LS180 melanoma cell line, and then we basically are able to keep those constructs very identical, and then we are able to create a population of the construct which we can use in further application where we do the treatment, and then we don't have problems with a different shape and sizes, where are actually the limitations or the factors which you need to very much consider when you do the experiments based on the 3D constructs. So this is another example where we can actually, when we use another type of gel and primary human hepatocytes, and also grown in the hydrogel. This is a 2-component hydrogel, but basically very easily you can create an extremely identical construct. So this is a big plus when you do the toxicological experiments. when you work with different cell lines, you also will need to think about the different adaptations. What you have to do here is actually example of a single cell suspension construct created out of different subclones of the same cell line. So the small difference in the subclones were connected to the malignancy level of those sub cell lines, but you can also see that they are having a huge effect on the shape and an ability to create an 3D structure. I also would like to say that using the ClinoStar effect, you can actually work out with constructs which are not extremely nice or not very nice for the repetitive studies. So, for example, here is the INS1E cell line grown for 7 days, but if we actually grew them for 21 days, we were able to utilize the clinostatic effect of a clinoreactor and actually create an identical shapes out of those initial irregular constructs. When we do those shapes, it's like, okay, it's nice, but what it gives us? So it gives us actually an functionality. This is what we are interested in. So here we have a few examples of different measurements we have done based on the HepG2C3A cell line. So it's an hepatoma cell line, immortal cell line. So very, very simple model. But you can see that if you start to grow those cells in the right environment, then you will get a more physiological-like response. And what is very important, you can also do repetitive and long-term studies without need of trypsinization and with full control of the biomass you have. And that is actually an example of a picture we got out of our construct. So you can see how the cells look like when they are grown in the monolayer and how nice and structural they became when they grow in the 3D manner. And basically, and structure and cellular communication, this is what makes a tissue. So this is what we actually wanted to mimic. So now I would like to very quickly go through some examples of use of our system. So why researchers are actually using it. So for example here, Dr. Agnieszka Rybak from Max Zulbriger, her goal was to try to see if she can remove the gel tracks out of her neuronal construct, because the gel tract is the not completely reproducible component. It's a natural product. It's basically like fetal calf serum. You are very much batch dependent. So they were actually growing construct based on the traditional method with use of gel tracks, and then on a method without using the gel tract in our system. And actually they have shown that they are able to create a very nice and functional neuronal construct without use of gel tracts. And here we have another example of growing breast cancer cell lines from Daniela Grimm from University of Magdeburg, where they actually were not very interested in the nice shape. They were interested in the conglomerate of the cells, but they were able to actually achieve a very nice functionality they were looking for. So another example is from Dr. Simone Sidoli from Albert Einstein from New York, where he actually was able to keep alive patient biopsies in our system. So basically this proves that the system is very nice and very nicely in supplementing and construct with nutrients. So basically you are able to keep a very compact tissue alive for a quite prolonged period of time. And this is an actual example which probably our Slovenian colleagues would like to see. This is something which is happening actually in Ljubljana. This is a group of Professor Bojana Žigura and Martina Stemper has been working in on actually different aspects of genotoxicity with use of the ClinoStar. And here you can see some of her research where they were actually creating a beacon population of the spheroids, and then they were actually analyzing how the constructs react depending on where the cells are. So they were analyzing the outer rim and the inner core of the constructs. So I think Bojana has a whole a pipeline how they actually can use the system, and that's very important. That a system by itself, it's not a solution for everything. You have to know how you will implement it into your analytical pipelines, into your production pipeline. So this is actually a very nice example how National Institute of Biology is doing it right now. And then here is actually some of the examples of already published examples from Dr. Martina Stamper, where they were actually using the construct created in a ClinoStar system to assess genotoxicity. And another example of use of our system in cancer research is actually a creation of the model for the small cell lung cancer model, or the model based on the H69V cell line, where different subclones can be drug resistant or chemosensitive lines. So that's also very important to have uniform sizes to be able to access this, and that was what we were able to do in Potchefstroom. And then here we actually use this model to actually work with compounds from the natural plant Saturnalia sativa, and then we basically use the model as a ready pipeline to assess the anti-cancer properties of this drug. And then something which was a recent addition. So you know, when you do the cancer study, you quite often would like to do the xenotransplantation, and when you do the xenotransplantation, you would like to do palpation checks after how the tumor grows after implantation, and that's very important to have uniform sizes in the start, and it's also very important to have a nice acceptance level from your inserted construct. So here is actually an research of Dr. Wienen Pfeiffer, who is actually using the system to create a material for xenotransplantation. And a bit a different example where you use the system to create constructs which will represent a neuronal tissue, and you use this one as a host for the virus infection. So that's also published data from Australia, from Dr. Romo Stewart. You can see this. It's actually very interesting that some variants of COVID virus are not very clinically aggressive, but they are actually showing a quite high affinity to stay or invade a neuronal tissue. That's actually work in progress. So they have published first article, but I think they will see much more and very interesting materials coming from Dr. Romo very soon. Yet another example where we use, where the system has been used to create constructs made after the long tissue or long cells. So this is an example from Medical University of Innsbruck from Professor Doris Wilfangstetter, where they actually also studied the COVID infection, and they can actually image COVID virus implementation into the organs. So that study is also in a preparation for publication, and I would say that you can also find a webinar on our webpage where this research is described in much more details by Professor Doris. And then yet another one. I know that I am going a bit close to a time end, and I just would like to show you that Professor John Wu and Dr. Carlos Pinzon has used our system to create a synthetic embryo. So a construct made out of different stem cells or induced stem cells. So they took the bovine fibroblasts, then induced them to pluripotent stem cells, and then created a very nice early stage look like embryo or blastoids. So actually here is a very nice image so you can see the blastocysts, which are the native structures created by organism, by cow, and then blastoid is actually synthetically created by Carlos. So they are basically of the same shape and the same functionality. They are difficult to distinguish. So what I would say that just to summarize, that I bombarded you with different examples, but what I would like that you remember is that we actually have an environment with a low shear force. We can do the long-term experiments with long-term treatment, and we always focus on the functionality and in vivo functionality. And right now, ability of adjusting a different oxygen level gives you a more flexibility to adjust to a different specific tissue environment. So basically, that was it from my side. And I would say I would be very happy to take any questions. And if you are curious, you can always visit our webpage. We have quite a lot of videos, a lot of protocols. I would also say that people from Slovenia could visit probably National Institute of Biology, where kind of a few machines is standing and working really, and those are the real users of the machine. Thank you very much.
Thank you for a great presentation and your work. It's really impressive. So I see some questions in Q&A section, but we will answer it after the next talk. So our next speaker is Dr. Devrim Pesen Okvur. She's a co-founder of Initio CelVivo. Dr. Pesen Okvur obtained her PhD degree in cellular and molecular physiology from the Johns Hopkins University. After her postdoctoral studies at the Karolinska Institute, Royal Institute of Technology, and Albert Einstein College of Medicine, she joined the faculty of Izmir Institute of Technology in Turkey, and her research focuses on design and fabrication of organ-on-chips that mimic the cellular microenvironment, and also on biophysical and microenvironmental control of metastasis. So Devrim, the floor is yours.
Thank you. I assume you can hear me and see me. Thank you for the invitation. I'm gonna talk about microphysiological systems as disease models. This data I present is some of it our own work and some of it is from our partners. We focus on cancer. It's the leading cause of death worldwide. And of course, we want to develop new drugs for this. However, drug development is a painful process. The failure rate is really high. It takes at least 10 years to bring a drug to the market. And meanwhile, the R&D expense is really extraordinary. Interestingly, the 30% of this R&D expense is on the discovery and preclinical stages. And those are actually the weakest links. And the candidates found in these stages, unfortunately, go to the clinical stages. If you look at how we see our bodies in vivo, it's complex, it's 3D. However, we are using 2D to study this complex microenvironment. And that's why we fail. If we compare the human body and the 2D systems, 3D and animal models, we can see especially 2D models are very limited. And if you look at from the perspective of the immune system, we don't really have a really good in vitro system. Even the animal models don't give us the same results because it's a different immunology. Now, okay, we can use 3D cell culture, but going beyond, we can actually do organ-on-chips. And this organ-on-chip technology is based on microfluidics. And the important part is we have control in space and time. If we can control the space and time at sub-millimeter levels, we can actually mimic the human physiology, the microenvironment. And since we can use human cells, tissues, biopsies, etc., we can offer replacement for animals. And this is actually very critical. Why now? For the first time in history, FDA allowed the clinical trial to begin based on data acquired using organ-on-chips or microphysiological systems, and no animal testing was required. Also, USA and many countries have begun to pass laws that support alternative methods. So they are saying if you can do an experiment without using animal testing, you should choose that method. What do these things look like? These are videos showing what the MPS or organ-on-chip is. It can be in different formats, multi-well plate or slide format. Basically, there are different compartments in the system. And you can load your drugs, cells, gels, depending on the design of your experiment, and use your cell culture lab and do your experiments. These are also compatible with robotic liquid handling systems, so you can have high throughput. I want to give some examples of what we have done with these systems. First one is about the interaction between cancer cells and macrophages, an important component of the immune system. The model was that these cells secrete molecules, talk to each other, okay? But we didn't think the data was really supporting that in the literature. So we said, okay, how can we test this? First, we had to show that these cells can actually do invasion, chemotaxis. And for that, we use one of our standards, IC chip. There are 3 channels here. And in the middle channel, you can load a matrix, the gel of your choice. And at the bottom, you can load medium with a chemoattractant. And at the top, you can load your cells in medium that doesn't have the chemoattractant. And in time, you can actually see these cells move towards into the matrix towards the side that has the chemoattractant. Just to clarify, these channels are connected to each other. So if something is present in one channel, it will diffuse to the other channels in time. And this is actually important because then you can have a gradient, and cells respond to gradients. So we show that the cancer cells and macrophages do actually invade in response to serum, but we want to know how do they react to each other, okay? And then we designed this new chip where we have a middle channel, but the middle channel is a V-shaped. So on top, the 2 sides are closer to each other, and at the bottom, they are far away from each other. And we confirmed the diffusion coefficients experimentally and with simulations. But the nice thing is when we put the cells here, cancer cells and macrophages, we did see invasion by macrophages, but not the breast cancer cells. So if any of these cells is secreting something, it's the breast cancer cells. We did other classical experiments like ELISA and lifestyle imaging, but I'm here showing only the chip data. So we came to the conclusion that these cells need to be in close contact for us to see an effect of the EGF molecule secreted, not secreted actually by the macrophages. So here we tested different gel compositions like collagen and matrigel, and we observed the culture in different days. In collagen, we saw an increase in migration when the cells were co-cultured, because collagen is not a rich media in terms of growth factors, there is none. So the only source of EGF was coming from macrophages. But when we do the same experiment with matrigel, which already has EGF in it, we saw actually a reduction in the migration. And this makes sense if you go to the biochemistry. The receptor for the EGF is EGFR, and if you have too much EGF, the receptor gets shut down. You have a negative feedback loop. So we see the biochemistry at the cellular level using the organ-on-chip system. We also wanted to know about invasion, and here we look at the tissue-specific metastatic potential of breast cancer cells. So this is our classical control experiment. There's chemoattractant versus not, and we should see a difference in invasion. Now, in the middle channel, this time, it wasn't just a gel, but we had cells from lung, liver, and breast. And when we do that, we can see on top, you see an invasive cell line 231. They invade. At the bottom, you see MCF7 non-invasive, and they don't invade. But if you look at the top panel again, we have lung, liver, and breast. Here we see nice invasion into lung and liver, but not breast. If you go to the clinic and look at what's happening in the patients, we see metastasis to the lung and liver, but not breast. So we see what we see in the clinic on the chip. We also confirmed it with specific cell lines that are specifically going to lung or bone with microenvironments, and these also confirm our results. Another event happening in metastasis is extravasation. For this one, we changed the design a little bit. So we need a blood vessel interface, basically. And I'm going to show you images to orient you. This is the top view. So we have the green signal. Those are the endothelial cells. And then we have the side view. So we have more endothelial cells in the middle where we have access to the matrix. Remember those openings between the channels, and not many cells on the PDMS part, but that is irrelevant. So, and then we should see a nice side view showing that the endothelial cells are coating the channel. So these are the fluorescent images. And in this video, you can see actually, yes, we have successfully coated the channel, and we see a lot of cells where we have the matrix interface. But we want to study extravasation. So we need to have not only structural, but also functional blood vessel. And we do this by testing permeability. So when we have endothelial cells, we have decrease in permeability as expected. And when we add the cancer cells into the flow channel, you see the red dots here. One of them is still in the flow channel, and the other one has extravasated into the matrix side. We can actually do this in live imaging. So we have here the green endothelial cells, and the purple are cancer cells, and you will see them actually do the extravasation. So they move, interact with the endothelial cell, and then cross the barrier. So these are nice images, but we have to quantify this, and that's what we did. Again, looked at the different microenvironments, and we saw what we did see in invasion. Breast cancer metastasizes to lung and liver. We did that with the specific microenvironment, specific cell lines. We also did this study with hepatocytes, liver cancer cells, because in the clinic, liver metastasizes to liver. So in our chip, we should see that, and that's actually what we saw. Here, the liver cancer cells preferred liver to breast and the bone microenvironments. We also collected CTC breast from breast cancer patients and looked at their metastatic preferences. So this is a small study of 30 patients. Now we are really close to getting the results on what actually happened in breast cancer patients, and we will compare our on-chip prediction with the clinical scenario. So going back to immunology, we wanted to look at infiltration by immune cells. Then we have a different chip for that, IC flow chip, where this time we have matrix on the side channels, okay? And in the middle, you can have media, for example, with immune cells. So we had 3 different configurations: cancer cells, normal cells together, or one side having no cells. and then we put the macrophages in the middle and see where they go. So here we see that macrophages prefer cancer cells over normal cells, which makes sense because all the secreted factors with the cancer cells and versus normal cells. And we can adapt this system to other immune cells, monocytes, T cells, etc. So, and we can study hot and cold tumor microenvironments. We can also use tissue. For example, here, what we did was dissect a mouse brain and take out punches and looked at the viability. Can we keep the tissue alive? Yes. And when we add endothelial cells to make a blood-brain barrier, do we see selective permeability? And yes, we did. And we actually saw the endothelial cells going into the tissue and making connections, which was really exciting. So we also looked at invasion towards lung tissue, in this case from mouse. But here we have an open top chip, which is much easier for handling. And we show that cancer cells invade much more when there are differentiated U937 cells. This is also what we expect to see based on the literature. So we can look at co-invasion and the effect of immune cells. Talking about drug development, we can see how cells respond to drugs in a 3D, but also dry culture environment. So if we compare a monoculture and we use different drugs, we can say, okay, both of them can kill the cancer cells, but it doesn't tell me anything actually. I need to know what's happening to the other cells. So by doing the triculture in 3D on chip, we show that this new drug candidate can actually kill the cancer cells, but it kills the normal cells almost 60% less. We can also look at anti-metastatic drugs. AMD 3465 is an antagonist of CXCR4 CX1212 axis. So this is important in metastasis and figuring out where the cells are going to metastasize to. And this drug does not inhibit proliferation, but we show in our system that it can actually inhibit invasion, which is really nice. We can also have multi-organ on-chip studies. Just to remind you, we are trying to go from really to complex models. And here we have a specific system where we can model the different organs using a scaled compartments. So here we can look at, we can generate PK/PD data, look at what happens to the drug and what the drug does to the cells. So then you can use this for downstream simulations. The thing with the dose response is it is important, but to determine it, the drug dose response is very time consuming and resource consuming. You have to test many different concentrations. Also, you come up with the in vitro data having micromolars, okay, mass per volume to mass per mass. So a lot of hurdles are on the way. So we said, okay, how can we do this better? We have this DR chip, okay? This is enabling us to simultaneously test multiple drug doses and combinations. So here we can load the drug on one side of the chip and it will diffuse in time. So we will have different drug concentrations, all possible in one chip. And we can also do drug combinations, loading one drug to one side and another drug to the other side. And there will be areas where we have both the drugs at high concentration, only one drug and none of the drugs. And we show that the simulations are in agreement with the experiments and we can get viability data using microscopy as well. This is nice because you reduce really the number of organ-on-chips to use. Even if you do this drug studies in the organ-on-chip platform, you have to use many chips because one chip, one drug concentration. So it will really cost a lot. So by using the DR chip, you can reduce this. And also the time costs and people costs because it takes people to run the experiment. And the chip is suitable so you can test small molecules, even agrochemicals. So I think I tried to give you an idea of what you can do with the organochip or microphysiological systems. You can study distance-dependent cell-to-cell interactions, many steps of the metastatic process, starting with invasion and followed by intravasation, extravasation, and so on. So it's really important that we use 3D multiculture systems. This way we can have, because of the advantages of the organ-on-chip system, single-cell resolution, and we can have real-time data. And we can actually focus on the drug dose response. By using animal and patient samples, we go towards precision medicine. And I think these organ-on-chip platforms are really useful tools for immuno-oncology studies. Thank you. If you have any questions, you can just email me and visit our website.
Thank you for impressive work and presentation. So now we have this question and answer session. So I would like to ask everyone, if you have any questions or comments, please write it on chat. So we have one question, I think it's for the first speaker about ClinoStar. So Sveta Rani is wondering if you are growing the cells for 32 days in spheroids. So did you check for apoptotic or senescent cells at the core?
Yes, that's a very nice question, and I would say we do. And if I could share one slide. All idea about an clinostatic effect is actually to be able to create an create environment where you can actually grow a more sizable construct with less technological hinders like a necrotic course. But I have to be also very honest, you know, quite a lot of people which study cancer biology, they are actually interested in having the a necrotic course on the constructs, because this is how you actually recapitulate yes, I will I just go
the next question.
yes, but the point is that using this active diffusion with an low shear, you are able to utilize this non-active vascularization on. You basically use the diffusion as a non-active vascularization system to get your construct supplemented with what is needed. And here is actually a slide where it shows a comparison of creation of an organoids with the use of a Pascal protocol or a standard one, or use of the ClinoStar, and the second condition where you actually put the construct in day 6 to a ClinoStar. So you can see a very nice reduction in the necrotic core in those constructs, and very nice functionalization. That's actually example from a laboratory from Switzerland, from Lausanne, from Professor Begini. And I think this is coming, this part of the first publication. So yes, we do check, and we have published methods where you can do the sequential disassembly of your construct of your spheroids or organoids. So you can do it semi-mechanically, or you can do it by digestions and do the flow cytometry, and then check the viability. But we are always connecting this with a functionality. So what you have as a functional outcome, what you would like to see. And we have grown our construct much longer than 32 days. We were actually growing them for over a year with preservation of the functionality and viability. Can you explain the advantage of the low shear stress versus high shear stress in the 3D cell culture? So if I can say it shortly, it's a bit like water. Water is needed for us, but if we have too much water, we don't really like it, and we can drown. So in some organs, they do experience shear force, but they do not have it too much. And I would say we had a publication about this, how much you can actually give to the cells and how much we are giving to them when we are using a technology to keep them alive. So our in vitro models are needing a flow or shaking or wave creation. So if you have an orbital shaker, you will create a wave and you will use usually something like 60 rotation or even higher, and then you will create an unphysiological shear. So this will basically affect your results, affect the how the cells communicate, how the cells the surface surface cells react. And it also has troubles with when you create the construct. So quite often, then you have to use a gel to kind of protect them from the shear. But when you start to use gels, you also diminish the diffusion. So it's a quite open and long discussion. So you would like to keep it at physiological levels, and our models quite often keep them in an unphysiological level, and that's not good. All right, I don't see.
Okay, so thank you. Yeah, so I also don't see any further questions. Is there any more questions? I have one for Devrim. So you mentioned that you also do 3D co-cultures in those systems. So can you just a little bit explain, like, do you do 3D models before and then do co-culture, or you mix before on the chip? So how does that work?
So we mix them at the beginning. We have tested, classical normal and cancer cells plus immune cells. We have had endothelial cells, fibroblasts. There's some data I haven't shown you, but we can do like 4 different cell types, and you need to tweak the medium to use and the different gel types. Some cells can be quite active. They can pull the gel. So we changed, if you're using fibroblasts, for example, we can use like fibrin, collagen, but we do start at the beginning. We mix them at different ratios, depending on the microenvironment we want to mimic, like the different stage of the cancer. For example, you can have fewer cancer cells or more of them to look at the initial and later stages of the tumor development. So we do mix them in the beginning.
Okay, thank you. Are there any more questions? Like, you can also, if you like, you can also ask live. So raise your hand and ask a question. No.
There's maybe I just found the slide about the shear force. If Emmanuel is still interested. So here you have actually the publication from 2018 where we describe and how our system works. And because the cryostatic systems, they are quite often called microgravity bioreactors, but when they are used on Earth, they are not really microgravity. So we have a few values, but then here in the end of this packed slide, you have your answers about a shear, what we actually are trying to mimic or not exceed. So. And if you want to know more about this, just go to our publication. We describe them different aspects or different options of the of different system. How much you will have it. Yes.
Yes, thank you. So maybe I forgot if you mentioned. So I know that you are now also have these hypoxic incubators. So that you can mimic these hypoxic conditions. So maybe you can mention a little bit about that.
So yes, so we do have the machine is able to regulate the oxygen levels in the atmosphere of the chamber, but we are also now having actually, and this is commercially available. You can basically purchase it, but we also work in our lab on the systems where we have sensors inside of our liquid, so we actually can measure the levels of the oxygens which are really in a contact of with the contact with the cells. So normally you will say, okay, if I have a close to physiological level of physiological liquid, my media is close to the physiological liquid, so it's an 0.9% of salt or whatever, and we have a 37 degrees, then you will basically, by a rule of thumb, say, okay, I have around 40 times less of oxygen than I would have in my atmosphere. That's what quite often we do as an assumption, but then I would say, all right, it would be very nice to actually measure it. So in our system, if I can just, I probably showed you some because I showed the pictures. I just didn't talk about them that because it's not commercially available yet. We are preparing the units to be commercially available, and if I can share a screen, I can show you actually how they look like. You see those dots, the big dots in the cell chamber? They are actually sensors. They are oxygen and pH sensors, which are measuring the levels inside of the system. So I was going very quickly through this slide because of the time restraints, and I would say I'm very, very happy that you asked, because that's the graph which we show here is actually what really happened, not in the incubator chamber, which I said that this is like a 40 seconds, but what really happened inside the media, what happened, how you actually equilibrate the media in your culturing vessels when you have a medium which has been saturated completely in the atmospheric levels, and then how much time you need to actually equilibrate it to the level which you are set, which you have actually set your incubator for. So that's a very important fact. And the other thing is also what the gradient will be. You will be having of this oxygen, because in the standard systems with no movements, you will always develop a gradient of the oxygen in your media. In the systems with movements like shakers or like our ClinoStar, you will actually have an uniform levels of the oxygen. So you basically don't have the dissonation at the time. So it's a nicer for a model, and then you can create a zonation in the tissue, not in your surroundings, and let's say like this. So soon, yes, you will be also able to measure those one. We'll be able to commercially offer that one.
Okay, great. So okay, I don't see any more questions. So I think we can slowly close the webinar. So I would like to thank the speakers. I learned a lot, and I think all those models are very useful to study the cancer, and specifically if we are talking about our cost actions. So they're very useful also to study immunotherapy and response to immunotherapy and toxicity of immunotherapy. So what I see, it's a great potential. So thank you, speakers, for your time and also willingness to participate. All the listeners that they took their time to listen to these great talks. And also I would like to thank the people who made this webinar possible. So Melanie, Eva, Rosalinda, Hanne, and Devrim. So stay tuned for our activities, and if you're interested, and if you're not a member of our cost action, please check our website and become a member. And yeah, so this is it, and thank you for your time.
Thank you.
Thank you very much for the invitation. Thank you.
Thank
Bye.
you. Bye.